So Eric is really a tough act to follow. I was just thinking I had some Dannon yogurt this morning. Eric, is my hair looking a little shiny? I'm going to keep adding that into the diet every day. We're going to jump back into the sediments now, and in particular sediments in the deep sea. And I'm going to tell you a story about a very interesting microbial partnership that is used to essentially consume methane that's fluxing up from the deep subsurface. And some of the tools that we've developed to try to understand this uncultured association. And there's a number of folks that I've acknowledged on this title slide here that are students and postdocs and collaborators, both from Caltech, UCSD, some folks at CAMRA, and Oak Ridge National Laboratory, and I'll try to acknowledge them as we go on. Okay, so I'm assuming that most of you in this room, a little feedback, should I stand back here? So I'm sure most of you in this room now understand and appreciate the value of various omics technologies in terms of understanding the diversity and metabolic potential of organisms in complex environments. But really, as Lee mentioned in his opening talk, the big challenge is, or Leroy, is trying to make this connection to their functionality and niche in the environment. In particular, how these organisms interact with each other to transform matter and energy in various habitats, whether it be the human body or a sediment community or a water column. It's really one of the grand challenges here, connecting the diversity of organisms and their function, and in particular trying to do this at the microscale that's really relevant to the organisms and what they view in the natural world. And so Dr. Azam so beautifully introduced this to us in the first talk today. So we know that most environments are not consisting of organisms free-floating in isolation, but rather they are part of well-structured communities, even in the oceans where we previously would have considered them to be very finely dispersed. And so in particular, when we're dealing with biofilms or sediment communities, trying to understand the spatial interactions of organisms really makes a difference in how they perceive the natural world. Okay. So the way that we've tried to approach this problem in my lab is to take some of the technologies that have been developed in the omics field and molecular tools and combine it with techniques that have been championed by the geosciences, in particular those types of technologies that allow us to query the geochemistry and stable isotope distribution at the microscale. And in particular, I've been using secondary ion mass spectrometry, or SIMS, or nanoscale SIMS, to actually query the isotopic or elemental composition of individual cells in the context of mixed complex microbial communities. And so really working at the interface of imaging the microorganisms in their natural habitat, looking at their geochemical signatures and metabolic potential through these molecular biomarkers is really allowing us to get a much richer picture of what's going on in a variety of environments. So the system that I've been working on for a number of years now is in the deep sea in places where methane is actively adducting towards the seafloor. These are called methane cold seep environments. And in these environments, this organism here, which is a symbiosis between an uncultured archaea and sulfate-reducing bacteria, shown here in the green on the outside, form a cooperation essentially to turn methane into CO2 coupled with sulfate in seawater, producing hydrogen sulfide. And these guys are important not only because they essentially control the flux of methane from the deep ocean to the atmosphere, but also because they're essentially the engineers that structure these very productive communities on the deep sea floor. So many of you are probably familiar with hydrothermal vent environments as being these oases of life in the deep sea. Methane cold seeps are analogous kinds of environments, except for the hydrogen sulfide that fuels these chemosynthetic ecosystems is all derived from this microbial partnership of organisms that are turning methane into CO2 and carbonate on the seafloor. So another reason why this particular partnership is interesting is just looking at the equation here of how they're taking methane combined with sulfate to CO2 and hydrogen sulfide is really an energy-poor process. And so these organisms essentially are living at the energetic edge, and they're sharing these meager resources in a very effective way to catalyze a process that is, at this time, thought to be the dominant way that methane is sequestered in the ocean. All right, so looking a little more closely at what we think is going on with this partnership, again, these are uncultured organisms, and so a lot of what we've learned about them has been inferred through indirect types of approaches, is we have, we have our methanotrophic archaea here shown in red. We know that it is the primary methane oxidizer, and it's converting this methane to an as yet unidentified electron carrier. And then we believe that this source of electrons then is passed to the sulfate-reducing bacteria, leading to the reduction of sulfate to hydrogen sulfide. And the secret to this success is really keeping this electron transfer intermediate at very low partial pressures, and this allows the process to continue to drive forward. And again, the amount of free energy that they gain from this process is very small, and both these organisms have to share these resources, and so as a product they grow very slowly, so on the order of, on a good day, a 3-month doubling time. Okay, so challenging system, but this is really how most of the natural world works, especially in deep subsurface types of communities. So life is sluggish. Okay. So we've learned a lot by just applying sort of molecular inventory studies of these communities in terms of the diversity of organisms that are likely catalyzing this process. And to date, there are 3 major lineages of archaea, all uncultured, that we believe are coupled to this methane-oxidizing symbiosis. And they're all called ANME, which stands for anaerobic methane oxidizers. They're not very creatively named. It's group 1, 2, and 3. But they have very different morphologies. So the ANME1 tend to be rod-shaped. The ANME2 and ANME3 tend to be more sarcina-shaped. And you find them either in partnership with sulfate-reducing bacteria, or sometimes you find them by themselves in the environment. And you see these mixtures of associated and unassociated all existing within the same ecosystem. We also see diversity in terms of the syntrophic partner that's associated with these methane-oxidizing archaea. In this case, I'm just highlighting 2 major groups of sulfate reducers related to the desulfosarcina and the desulfobulbaceae that both form associations with a specific methane-oxidizing archaea. So this is just using fluorescence in situ hybridization and looking at the range of diversity that you see in the environment. And so as ecologists, our questions are, we see all of this variation existing in these habitats. What does it mean in terms of the overall functioning and efficiency of these relationships? Does the partnership matter? Does the spatial arrangements matter? How close do these things need to be to one another in order to efficiently oxidize methane? And so these kinds of questions are really best addressed using an approach that allows us to study them at the microscale and look at them in the context of their natural environment. So to take advantage of some of the technologies that have been developed, mostly in the material science and geoscience fields. We've been applying secondary ion mass spectrometry to get information about the isotopic and elemental composition of individual cells that are recovered directly out of marine sediments. And so in this particular case, we can set up incubation studies of sediment that we've collected from the deep sea. We can feed it methane, and we can give it an isotope label. In this case, we're feeding it N15 labeled ammonium and ask the question, are cells that are living on their own as active in producing new proteins as ones that are in association with each other under conditions that are supporting methane oxidation? So if we look at this diversity of different cell types, are all of them active at the same time under the same types of conditions, and if not, what are the differences? And so to do this, we would apply fluorescence in situ hybridization to identify cells and then analyze them directly to see if they had taken up this N15 label using secondary ion mass spectrometry. And the way this technique works essentially is you can focus a primary beam of ions. In this case, we're using a cesium positive ion beam. Essentially, these ions get implanted in your sample. Secondary ions are excited in your material and then are sputtered from your sample matrix and then are collected through a mass spec. And so you can get information on essentially the abundance of 15N to 14N or other types of elements. And the secret here is we can focus this primary ion beam size down to 50 nanometers or 10 microns depending on the instrument. So you can actually get subcellular information in some cases of individual cells. Okay, so this is sort of a nice cartoon showing, the destruction of a single cell here with a primary ion beam that came from a review by Mickey Wagner a few years ago. And so I'm jealous actually that I didn't actually have this cartoon in one of my publications, but the sausage is essentially being stripped of its macromolecules, and then this information then is converted into essentially an ion map that shows you in this case, the elemental distribution of either sulfur or nitrogen ions in the cell. And so we can apply this not only to fish stained cells, but also to TEM thin sections. And you can see the spatial resolution is quite remarkable when you get a very flat surface, even to the point where we can start to see intracellular inclusions in these organisms. So this level of insight and really has given us unprecedented information about the structure of individual cells and their influence spatially on each other in the context of a mixed community. Okay. All right, so we had this question of whether single cells versus cells in association were active under conditions supporting methane oxidation. And so we set up, again, these incubations using N15 labeled ammonium as a proxy for new protein synthesis in the cells, and analyzed a number of these different organisms here. And I just wanted to show you an example of an ion image that was collected with the nanosims here. This is the fish image of a dual species aggregate that's in the process of dividing. And this is the corresponding 15N, 14N ratio image of that cell after incubation with ammonium. And the warmer colors represent more incorporation of the N15 label. And so what we see here is that there was new protein synthesis throughout the aggregate, so background is black, but that the area where you see most protein synthesis is actually occurring at this division plane between these two multicellular aggregates. And so it suggests that you're not having a single cell that's released and then recruiting a new symbiotic partner, but rather these organisms are wanting to stay together in sort of physical association and then pinching off and dividing and then forming a new aggregate and going off. Okay. So this is a summary of some of this data now. Each individual point represents a paired set of analyses collected with the nanosims. This is natural abundance C13 and atom 15N incorporation here on the x-axis, and so more incorporation of the label is along this way. lighter values in C13 represents more incorporation of isotopically light methane, so naturally assimilated methane. And what we see is that these organisms that were found in paired association with one another rapidly took up the N15 label in these incubations. And if we compare this now to those that were found on their own in these same incubations, we see very little uptake of both the methane-oxidizing archaea as well as the sulfate-reducing bacteria and other bacteria in the samples. So when you're stimulating anaerobic methane oxidation with sulfate, being close together in these partnerships really makes a difference in terms of the overall activity and growth. Okay. All right. So we wanted to go a little bit beyond just looking at sort of the basic metabolism of methane and sulfate utilization, and wanted to understand how these organisms were physiologically adapted to the environment. And to do this, taking a metagenomics type of approach is a good first pass to gain sort of insight into what their metabolic potential might be like. And so as I don't have the Banfield Army working behind me to really deconvolve these very complex sediment communities, and so instead we decided to take a more directed approach to try to selectively extract out the aggregates of interest to the exclusion of all of the other organisms in the environment. So by dealing with this lower complexity sample, we were able to get more targeted information related to our methane-oxidizing consortia. And so there's a number of different ways that you can enrich for organisms in the environment. The way that the approach that we used was essentially to try to magnetize our multicellular aggregates by using a 16S targeted ribosomal probe, and then using an antibody type of approach that's targeted to the fluorophore that we stain the organisms with. We can essentially attach paramagnetic beads to those organisms and then gently extract them out of the sediment. So the nice thing about this approach is those hitchhiker organisms that Paul was really trying to get rid of, we actually want to hold on to because we want information about the physically associated partnerships that are occurring naturally in the environment. So these are archaea that were stained with the probe, and then you see stained with DAPI here, all of the other types of bacteria that were physically associated. So by looking at this now enriched population, we can get information possibly of new types of organisms that are in these associations. And then we can use this information for targeted metagenomic sequencing, getting information about potential function. And more recently, we've been using this metagenome as a reference library to try to interpret environmental proteomics data. Okay. So just to show you really quickly a little bit of data related to our methane-oxidizing archaea. And so there's been a few different metagenomic studies. And in this initial study that came out of Ed DeLong's group, they found that with the ANME1, all of the genes involved in the 7-step canonical methanogenesis pathway were actually present in these methane-oxidizing archaea, shown here. And so we wanted to see if our ANME2s had very similar types of capabilities. And so indeed, when we looked at our partial metagenome, we found all the evidence of all of the same genes present in the ANME2. So these organisms appear to be very similar phylogenetically and evolutionarily to methanogens. And even more satisfying, if we looked in the environment with proteomics, we see that many of these genes are expressed. Okay. But in addition to this, we also found evidence of nitrogen utilization genes, and so we wanted to look a little more closely at this capability within the methane-oxidizing archaea. And in particular, we found evidence for potential for nitrogen fixation by these methanotrophs. And if we look phylogenetically at the distribution of these methane seep NIF genes, we find that they are distinct from everything that had been previously described. And because of their unusual positioning in this tree, it's very, very difficult to ascribe who these genes are associated with. And so we set up a series of incubation experiments again, this time using N15 labeled dinitrogen to ask the question of whether or not our methane-oxidizing archaea were capable of fixing nitrogen in tandem with methane oxidation. And so, again, we used fish, we identified the organisms, and then analyzed them by nanosyms. And this was work done by a graduate student in my lab, Andikis. And so this is just one example here where we targeted a methane-oxidizing aggregate with fish, and then it's the corresponding fluorescence in situ hybridization image looking at the ratio of N15 N14. And we see as we drill down with depth into this aggregate, we see more and more incorporation of the N15 label that appears to be associated with the methane-oxidizing archaea shown in red. And so if we look at this more closely, so the red bars are related to the methane-oxidizing archaea, and the green bars are associated with the sulfate-reducing bacteria. So although we see a lot of variation for individual cells within each step, we do see that over time the archaea appear to be more enriched in that label. And the interesting thing is that some of the label is actually being shared with the sulfate-reducing partner. So we think that the archaea are the primary fixers. Some fixed form of nitrogen is being passed to their sulfate-reducing partner. And to compare this to the kind of information you would get if you just did one single bulk analysis, it gives you a much more rich picture of the diversity occurring within individual cells within this cluster relative to that in the shaded region here. Okay. So looking at sort of the take-up of various nitrogen sources with under conditions supporting methane oxidation, we see that it follows a trend that's very similar to what you would expect based on the energetics of using these different nitrogen sources, where ammonium over time is utilized much more effectively than nitrate versus N2 and cyanide. And so, so it appears that they can use a wide range of nitrogen sources, but that the energy invested in using these different sources varies very strictly with the amount of energy put into using different forms of nitrogen in the environment. Another interesting component of this is that these environments are very heterogeneous and occur across very steep redox gradients. And so it turns out that where nitrogen fixation occurs is typically localized in very small spatial scales within the bulk sediment. And so if you don't know where you're looking, you might actually miss this important process that's occurring. And so here we see that at the 6 to 9 centimeter depth horizon, we see incorporation of the labeled nitrogen relative to the upper and lower horizons here. So if you've homogenized across this whole thing, you might lose your signal. Okay. All right. And I'm just going to end with talking really quickly about another project where we were interested in looking at the differences between the different sulfate-reducing partners, and in this case, the potential for niche differentiation depending on which association the methane-oxidizing archaea formed. And so we're looking at the desulfo-bulbaceae sulfate-reducing partner and the desulfo-sarsina partner. If we looked at their distribution in the environment, it turns out the desulfo-bulbaceae-related aggregates tend to be most abundant immediately below sulfide-oxidizing microbial mats relative to other types of seep habitats, and tend to be more abundant in shallow layers relative to deep, which suggests that there may be something unique about that habitat that's stimulating their growth. And so if we look at these environments, typically right beneath the microbial mats, we find high pore water nitrate levels. And if we looked at the genomes, metagenomic data, we saw potential evidence for nitrate utilization. So this was essentially a hypothesis that we could go again and test with SIMS. And so in this case, we added N15 labeled nitrate and looked at aggregates that were either in association with desulfobulbous, the DSB, or with desulfosarsina. And indeed, what we found is under conditions supporting methane oxidation, that the desulfo-Bulbaceae-associated aggregates were better at taking up that N15 labeled nitrate relative to the desulfosarcina. So this may provide a slight selective advantage for these organisms where they exist in nature. Okay, and so I'm going to skip this really quick. And so I'm just going to end by saying that we can use this combination of various omics types of techniques now and really go after very specific hypotheses and bring in these other types of technologies that allow us to begin to sort of pick apart the complexities of microbial communities. And I wanted to iterate what Professor Azam had said earlier today, that these imaging types of tools that really allow you to look at the spatial relationships between organisms are fundamental, I think, in understanding a lot of the diversity and metabolic complexity that we see in nature. And so we can now essentially drill down and ask very specific hypotheses. If you have a good question, there's a lot of different types of imaging techniques that you can use now to go after the functionality of organisms in the context of their natural environment without having to grow them in the lab. And so with that, I'll end by acknowledging my research group and in particular Abby Green and Ann Dekas, graduate students that worked on this project, Sean McGlynn and Josh Steele, postdocs, and a number of collaborators from UCSD and CAMRA, Mark Ellisman, Tom Deering, Shulei's son, folks from the Ion Probe Lab at at Caltech, Yungbin Guan and John Eiler, and then various collaborators from over the years. So with that, thank you.